when you studied cell structure. In Mumbai University, in Mumbai, Maharashtra, now all these places in CBSE, it has come in 6th, 7th standard. Cell structure, eukaryotic cell, prokaryotic cell. Cell structures and organizations are also being taught in MSc. So I asked a question that what was taught during your 6th standard and what is being taught in MSc? Is there any difference? Why it's being taught? Because subject might have not grown, but your mind has grown. Those days you were only taught to keep in your mind some image. Now you are being taught to imply that. If you know how to break the cell, if you know where DNA is located in nucleus or cytoplasm of mitochondria, if you know how to isolate these organelles and nucleus and other things, and if you know that how you can solubilize the membrane, plasma membrane, you will get your DNA out in the sap. Then if you know that how to remove the carbohydrate, how to remove the lipid, how to remove the protein, you will get the pure DNA. So why don't you write your own protocol? Believe me, my teaching was quite useful and I felt admired. I gave this question purposely in the exam, and she was the only student who wrote the protocol by her own only, but of course in a lot of distorted manner, and I gave 100% marks on that question. So what I am saying here, that when the crisis comes, evolution comes, which you need to understand. So the second point, then the vision has to be there, that why are you doing the such thing? You have to have the urge, commitment hona chahiye. I am going to do that. I can't escape from this problem has to be solved, and then determination and ability, of course, which I am repeating almost every single example I gave to you. You need to be capable. You need to develop the capability, and how it will come, we will see in my last slide. So in this case, where was the crisis? The crisis was the southern hybridization. I don't know how many of you know that. If I take 2 minutes to explain you, southern hybridization is a technique through which you detect the DNA by using DNA as a probe. What is probe? I am very good for giving the classroom, probably not for inspired lecture. I can make lot of your fundas clear. What is probe? Probe is a literal meaning. Probe is an element or a kind of compound or anything you talk about which you know everything. This is called probe. If you know if you know that this is, if you tag this finger of mine, that this is H.S. Misra's finger, this becomes a probe. If you throw this finger and it goes and binds to anywhere, you can find out that this person also has got a finger like H.S. Misra. So this can act as a probe. So probe is a molecule, an element or a compound about which you know everything. That's called probe. So what southern hybridization does here, that you take a DNA as a probe. If you are interested to identify a gene of your interest, you know the gene. Since you are 10 plus 2 students, so maybe I will have to go up and down, to and fro. Gene is a part of the DNA from which a protein is being encoded in your language, the simplest language. So you know the protein. Protein is a building block of our body, physiology, everything. Now protein is, the information of the protein sequence is present in the gene. That's a piece of the DNA on the chromosome. So if you want to identify that, I want to know that gene for this particular protein, you pick up the DNA, label it with 32P or anything, and then you search in the entire fragment of the DNA. You pick up the DNA, digest, fragment it to small pieces, separate on the agarose gel, and then you do hybridization. So hybridization you are doing, you are having a probe, and then you have got a target. So when probe goes and meets to the target, wherever it finds the complementarity, which I mentioned in the DNA structure, it will light up. So in southern hybridization, that is what has been existing for a very long time to detect the gene, isolate the gene. Now I am telling you the crisis, where the crisis comes. The technique has been working brilliantly, but at a time you can do only one gene detection. I want to give you a background. At a time you can do only one gene detection, and one complete experiment of the southern hybridization, it takes 5 days minimum. So for detection of one gene you need 5 days. For detection of more than 50,000 genes in the human genome, how many days you will take? Probably the entire career of your scientist. So that has been the crisis. Crisis was there that if you take 5 days for detecting one gene, and if you want to detect or study some 20,000 genes, how will you go about? So that was the crisis. Something thinking was there that can we get some technology. What was the inspiration, my dear? Computer chips. This was the inspiration of getting the microarray technology. I am truly sharing with you. You know the chips, my computer chips, I am 100% sure every one of you know. Everybody's mobile and computer will have such thing overloaded. So what was done? My computer chips were developed. So there they realized that you can decode million information on a small piece of millimeter size of the hardware. This was one thing. So inspiration, crisis, and then there was all these things to develop such technology. So what happens? Then in that case, automation through mechanical engineering and electronics was also contributed. I will come in details that what exactly this technology is or not. And computation through computer engineering. So you can imagine there is a contribution of physics, chemistry, biology, primarily electronics engineer, computer engineer, and many such things. When mechanical engineer, they all came together and then they contributed for development of the microarray technology. So this is all you talk about. We'll go next. This is just a schematic representation which I picked up again from the web page. I want to quote it because I want to comment at the end of this also. You see there is a spotting. So what in microarray technology, what is being done basically, let me tell you the structure. You take some microscopic slide, you have seen microscopic glass slide. You take up that size of some wafers, plastic wafers, high quality wafers or glass, and then you spot some 30,000 spots you have to make on that cover glass slide. Now you can imagine it. If you are asked to put with a pipette, if you are asked to put with 10 ml pipette, you will be making only one spot. If you are asked to make some microliter, you will be making some 30-40 spots. But when you are required to make some 50,000 spots for entire human genome coverage, what kind of precision you will require? You can't do by manually. So you need to have the automation. So spotting is the word is called. That you have the wafers, you need complete automation, and that automation required mechanical engineer and electronics engineers. They made the robotics, and all the spotting and everything is being done robotics. You read on microarray technology, probably you will get so much excited to choose your career in this area. In overnight, Affymetrix has become the billionaire because they got a technology, something which they could implement. And after that, once you do the spotting, you get all kind of spots made. Then you do hybridization. Now hybridization is basically very close to what people used to do in southern hybridization. So a lot of things borrowed from the southern. Then this is pure biologist work where you extract the RNA, do the labeling and purify it, and then you put together with the chip. This is the chip which they call it microchip, and then you mix together. You will do hybridization here. Then you do scanning to see where the spot. Without going so much detail, you will get this kind of structure. You can see here. I was struggling with the southern, where one gene probing used to take 5 days. Now 50,000 genes can be probed simultaneously in less than a week. And this all has come. What I want to tell you, can you go? This all has, go back. They will see what I want to tell next. So all these things have come. All these things have come because of the holistic marriage of physics, chemistry, biology, computer science and electronics. Since you all are growing, it's very difficult to convince a scientist like H.S. Misra, who is already 50 years old, that you go and interact with your colleague. Because I have already grown in the atmosphere which has been hostile. Keep the boundary, don't mix together. But at least I can don't take my word what I said just now, otherwise it will get percolated to you. But I am 100% sure that at this stage, if you acquire a sense, a responsibility, that no, I am not going to keep the boundary, I am not going to keep my feudal secrets and knowledge with me, it's not going to yield me anywhere. You will interact with the people in more holistic manner and then contribute in the better way. The next presentations, couple of presentations are how to make the money. Of course, here also there is a lot of money involved. Come in, that you need inside. You see this T7 DNA polymerase story, this inspires me like anything. Because I have met this man and what has been I am going to convey to you. How many of you know bacteriophages? I have time? I have time or what time? Break it. Oh, really? Let me finish it up. Okay, then I want to go my work. I will explain you all the story. How many of you know bacteriophages? Bacteriophages are the viruses which kills the bacteria. Let's put that. And it's being called PHAGE. Phage. Bacteriophage. T7 bacteriophage is one of the virus which can kill the bacteria, E. coli. The experiment was being done much before the Richardson, and someone came in the laboratory and did the T7 phage titer determination, that how many phage particles can grow on one cell of the E. coli bacteria. So they did plating. Next morning they come and then they know that the entire plate was clean. And person got doubt that whether I plated or I forgot it, I was thinking something else. He repeated the experiment couple of times. Every time he found the same thing. This was an observation. I want to correlate it. This was an observation. I think my session is interactive. Now you can ask the question. Let me continue on this. So what was there that now this was an observation. What was the reasoning done on this? We knew already that, as I have already explained you, that unless one DNA becomes the two DNA, one cell cannot become the two. So if you can produce million or 10 to the power 12 phage particles on one E. coli cell, you cannot make it unless the T4 phage, bacteriophage DNA has become 10 to the power 12 copies. Do you agree with me, no? If one phage particle is becoming 10 to the power 12, the DNA will have to become 10 to the power 12. This is a simple arithmetic. This was the reasoning done by Richardson, whom I am showing it here. And then he said that what can do that? The hypothesis was there that it's a DNA polymerase present in the virus which only can make several copies of the DNA. And he picked up this